for you. So I am formally starting and saying hello to all of you to another class of our medical biotechnology course, and I will going to complete the second part of the lecture about PCR, polymerase chain reaction. In the first part, if you are not in a position to recall that lecture, please go back and visit the channel and to see that video once again, where I have tried to explain you about the real big question behind the polymerase chain reaction. I have talked about what actually happened during the first, second, and the third and the fourth PCR cycle, and how and when specific PCR products are produced during the polymerase chain reaction. That was a real important thing to understand in perspective of polymerase chain reaction. The rest of the things are fine. Okay, so let me go formally to the traditional, some simple slides related to the polymerase chain reaction, its application and other things. Just let me go through those slides quickly because I don't need to spend much time, since we have covered the most fundamental aspect of the lecture, and that is that what is the mechanism of PCR reaction and who ensures, what is the factor behind the specific amplification of specific PCR product. And let me share my screen with you now, and I will be browsing my slides for you people from here on. But remember, the most important thing which you need to stay, which you should carry with you, is the fundamental question associated with the mechanism of polymerase chain reaction. Please go back and see the video once again in order to get that question completely. It's very, very important. So let me start with a formal reaction, because I have created those slides in the perspective of molecular diagnostics, because it is one of the very famous medical biotechnology based technique in which we can use PCR reaction for diagnostic purposes. And also, based on the current pandemic situation, we know that there exists a very famous reaction, PCR test out there, which our government and the rest of the world is using in order to accurately detect and confirm the patients with the coronavirus, to confirm the disease which we call COVID-19. And you know the difference, coronavirus is the name of the virus. The specific virus which is causing that disease is SARS-CoV-2. Okay, I have made a lot of videos related to that information, authentic information. I have shared certain research papers as well, and of course in Urdu, so that it can reach out to many people. About SARS-CoV-2 is the name of the virus, and the disease name is COVID-19. So they are the different things you should know. So let's start that how PCR reaction can be used as a tool for diagnostic purposes. And this is yet another interesting area of medical diagnostics. First thing first, we all believe that God has created life. Most of us believe on that thing, that there is a supernatural power exists which actually created the life. But until very recently, nobody knows actually how God has written the codes of life. We do know God has created the life, but how the codes of life was written was unveiled by the very famous Troika. I should not use the word Troika because I don't like two of them very much. The one is a very beautiful lady called Franklin. You might heard her name, Rosalind Franklin. Her X-ray crystallography work revealed the data which was used by the two famous scientists called Watson and Crick to create the three-dimensional structure of the DNA. And that was the very first time when we came to know that how the life is actually written. And it was written in terms of the codes with the help of 4 alphabets. Think how simple that thing is. All the complicated versions of life out there is written with just 4 alphabets. We need 24 alphabets languages, 24 alphabets in a certain languages to express our expressions, to communicate with each other. God has used just 4 of the alphabets to create all versions of life. And this is the most specific thing about life on the planet Earth. Because with all our abilities, with all our capacity, we never ever able to locate anything like DNA or protein anywhere else in the universe. We know what are the metals, non-metals, minerals are present in the other stars, the gases out there. We can detect everything. We manage to detect everything. We know that there are asteroids carrying tons of gold and aluminum, but we never ever able to detect any molecule like the DNA or the proteins. So I believe that when in the future, when our planet will be invaded by the aliens, they will be very much interested in DNA more than the gold and the dollars and our currency in our bank accounts, because this is something very special and unique to the planet Earth. So once we came to know that how life is written, the next step was obvious. Everybody knows what will happen next. And the next step is how to rewrite the language of life, how to rewrite the code of life in our own way, how to recreate the life or the things in a way we want. That's the next logical step. And we are in that journey right now. We are trying to change the codes in a way we want to create newer properties and better properties. So once we came to know about the DNA, we started understanding about the structure, and then we started using the presence of those DNA. By analyzing the DNA, we started understanding about the life itself and about the abnormalities in the life as well. For example, cytogenetic tests. These are the tests which we do to see whether there is any abnormal chromosome is there or not. Philadelphia chromosome is an example of a blood cancer. It is created because of the translocation of chromosomes. So in an abnormal situation, different fragments of the chromosomes merge together to make a smaller chromosome called Philadelphia chromosome. You can browse it further. But we can see that thing with our own eye under the microscope. Oh, the chromosome is not normal. So by looking at the DNA, we able to figure it out about the life as well, whether the life is going on nicely, normally or not. We can count the number of chromosomes to see whether it is normal or not. So the DNA is like a standard for us. If the DNA is intact, it looks normal, we can assume the life will be normal as well. The second thing, FISH, fluorescent in situ hybridization. In this technique, we send a probe. That is a very common word used by molecular biologists. Probe. Do not get impressed very much with the term if you don't understand. It's nothing special. Probe is just a fragment of DNA, a sequence of DNA which which matches with one of the target sequence in the cell. So that's what we call as probe. It can go and it can bind to a specific target in the cell. So if with that fragment, if you're able to attach, for example, this is the sequence of DNA. If you're able to attach a certain fluorescent dye at one corner, if you're able to attach a certain fluorescent dye with a sequence of a piece of DNA of your choice, this thing can be termed as a probe because it can go at the place of your choice. It can go at the place of your choice. And this fluorescent thing, this stuff will glow and it will let you know where that sequence exists. For example, if this is the DNA and this sequence matches with that particular part of the DNA on a chromosome, that sequence will attach itself here, and that glow will let you know where that gene will exist within the chromosome. So scientists started understanding about the location and localities of different genes within the chromosome. And there are different regions on the chromosome. Centromeric towards the centromere, telomeric towards the end. They started understanding where my gene of interest is present, or whether it is present or not also. And then there exist certain molecular tests like PCR reaction, which we are using nowadays to detect the presence and absence of a specific sequence of DNA in a sample. So the source of life or the codes of life are in use to analyze the integrity of life out there. Is used in diagnosis of infectious diseases, genetic identifications, and detection of genetical diseases, depending on the nature of the plan you are making to use polymerase chain reaction. Many famous infectious diseases can be detected with the help of polymerase chain reaction. HPV, hepatitis B and hepatitis C, HIV, all of them can be detected with the help of polymerase chain. All you need to do is to design primers which can specifically bind with a sequence of HIV, and that's all. If the HIV is present, the primer will bind and amplify. If HIV is not there, the primers will not bind and will not produce any PCR product. The same thing is happening with the coronavirus in case of COVID-19. The samples from the nose, from the throat, because the virus is supposed to be here, it's supposed to live in the upper part of the throat, especially in the first week. The samples were taken from the nose or from the throat with the help of the cotton swab. If the coronavirus is there, scientists should be able to get the RNA out of it. And coronavirus is an RNA virus. But that means you should be able to get or extract the RNA out of that cotton swab. Once you extract the RNA, it is converted into the cDNA. Once you have the cDNA, the game is simple and easy. All you need to do is to design a primer which can specifically bind with the sequence of the coronavirus, SARS-CoV-2. If the virus is there, if the SARS-CoV-2 virus is there in the patient sample, it will give off RNA. You should be able to convert them into cDNA, and you should be able to amplify the cDNA as well. The amplification of the product let us know that the person is carrying the SARS-CoV-2, and that is how we can detect the presence and absence of that virus in the sample. It can also be used in genetical identification, paternity testing, of course, and forensic, especially forensic, very, very important. In forensic analysis, you might remember the very unfortunate incident in Pakistan, in Kasool, a very famous Kasool case, where the suspects and the police and the state actually analyze the samples from all the neighborhood to confirm and to find the offender, to find the culprit. Very huge level. Everybody who is young at that particular age, everyone is tested. And the DNA of the offender is then matched with the DNA which was found on the clothes and the body of the victim at that time. So forensic analysis, the DNA testing techniques, including the PCR, is used extensively. And of course, the paternity can be confirmed very easily as well in forensic science. The polymerase chain reaction itself, I have explained it. I am not going to talk much on that part. The synthesis of a defined target of DNA in vitro, outside the cell, using the particular enzyme. It was discovered in 1983 by Dr. Carey. And the enzyme which was used to amplify the DNA was Taq polymerase. Nowadays, we are using another versions of polymerase, enhanced version of polymerases. For example, we are using PFU polymerases. Remember the terms, it's very important. Somebody might ask you. The most commonly used DNA polymerase in the PCR reaction is Taq. But the Taq polymerase is able successfully to amplify the DNA, but it does not carry the proofreading capability. It cannot fix the error. On the other hand, the PFU polymerase, another type of polymerase, is not only is able to amplify the DNA, but also if a base is inserted wrongly, it can fix that thing as well. It has an exonuclease activity, which means it can delete the wrongly inserted base pair and then can reinstall the right base pair there as well. So it has a proofreading ability as well. If you are just analyzing the presence and absence of certain thing, Taq polymerase is good enough. But if you are making DNA, if you are creating genes for cloning purposes, you don't want any error to be there. Otherwise, the function of the gene will be lost. So under those conditions, when you are synthesizing a gene, you should use polymerase chain reaction. Oh, sorry, you should use PFU polymerase as an enzyme. Also, the Taq polymerase, when synthesize a DNA, the Taq polymerase produces the non-blunt ends, which you might remember the term the sticky ends. It left few base pairs at the end and at the start of the DNA. But when PFU polymerase synthesizes the DNA, it produces blunt ends. Blunt end means like just without any overhangs of DNA at the end. The other difference. Why Taq polymerases are used? Why not we use human polymerases? Every cell carries the DNA polymerases. Every other plant is carrying the DNA polymerases as well. So why not we are using the DNA polymerases present in other animals? Why we have to use that enzyme which we extract from the bacteria present in the hot springs. You know why? The polymerase chain reaction, the mechanism requires you to boil the sample, to boil the DNA. If you remember, you need to separate the double-stranded DNA into single-stranded DNA. So in order to convert the double-stranded DNA into single-stranded DNA, you have to boil the sample. Boil is not exactly the good word. You have to take the sample up to 95 degrees centigrade, almost boiling. If you use human DNA polymerases or a plant polymerases or any other polymerase, it will be degraded because the protein structure will not survive at 95 degrees centigrade for sure. It will not work at higher temperatures. You know that. So the polymerase chain reaction depends and actually discovered when somebody found life in the hot springs. वो जो गर्म चश्मे होते हैं, somebody founds a bacteria present in the boiling waters. If a bacteria is able to live in the boiling water, एक बात तो पक्की है, the bacteria should have a DNA polymerase. And that DNA polymerase, क्योंकि bacteria ने जिंदा रहना है, it should divide its DNA as well. It should have a DNA polymerase which is strong enough to live in that temperatures. So the scientists at that time, they borrowed that DNA polymerase from those bacteria, and we are now using them to synthesize DNA in vitro with the help of polymerase chain reaction. Okay, the mechanism is simple. The things which are required: number one, target DNA. We should have a template. We should know the sequence, because if we know the sequence, we can design primers to specifically amplify a place of our choice. We use DNTPs. These are the nucleotides, deoxynucleotides, triphosphate, building blocks of DNA. The N stand for the 4 base pairs. Remember, if you want to show the 4 base pairs together in one word, you can use N. Actually, they are DATPs, they are DCTPs, they are mixture of DGTPs. So all the 4 base pairs, the mixture of all the 4 base pairs together, they are called as DNTPs. Then you need an enzyme, for example, thermostable enzyme, the enzyme which can survive at high temperature. Then you need magnesium ion, which is a cofactor for that enzyme. And of course, a buffer solution to keep the pH at a proper level so the polymerase chain reaction can proceed normally. At first, you need to heat up the sample. So the double-stranded DNA should be single-stranded. Then you lower the temperature so that the primers should bind themselves to the templates. हमने ये पढ़ा हुआ पहले, okay? I'm just repeating it. Once the primer binding is done, you increases the temperature to 72 degrees, which is perfect for the thermos or for the thermostable DNA polymerase to start synthesizing the DNA. The primer will start synthesizing the DNA after that. And you repeat that cycle again and again in the same way to get the amplified PCR products. And that which we have studied from the one single copy, you can have millions of them. And now you know it does not proceed in this way. You know what will be produced at the end of the first cycle. If you don't remember, go back to that lecture and listen to that lecture once again. You should know how many products will be there after the second cycle. How many products will be there after the third cycle. And then we start having like 2 million products in just 20 cycles. And it values increases exponentially after 17 to 18 cycles. This is actually the curve of Pakistani patients nowadays. The corona patients are increasing in this way. Anyways, so The application of PCR, it is used in the classification of organism, genotyping, molecular archaeology, mutagenesis. Mutagenesis is like creating mutations at the place of your choice in a way you want. You can replace A with the T, you can replace G with the C, or C with the A, whatever. You can do these things with the help of PCR. It is more accurately called as site-directed mutagenesis. Site-directed mutagenesis. It's involved in DNA fingerprinting. You can detect pathogens, genetical engineering, cloning, prenatal diagnosis. You can do a lot of things. It is used in basic research. It is used in applied research. There's a difference between these two types of research. In basic research, we detect things. We discover mechanisms, the natural mechanisms. We discover newer things which are responsible behind the functions of life. And in applied research, we use that information to create things, to have a better life. We create things. So when you're working in applied research area, whatever you are doing will go to the market in few years. And it is important for the countries like Pakistan. But if you are working in a basic research, basic does not mean less important, okay? All the big universities in the world, they are doing more research in basics. They're discovering phenomena, discovering new genes, their role, their participation, their roles in disease, their mechanisms. But for the countries like Pakistan, the applied research is more like logical. It will give you the product instantly. So it is used in molecular identification, in sequencing, in genetic engineering as well. So since the DNA is unique for every organism, so we might be able to find certain sequences of DNA which are specific for one organism. I'm giving you a very weird example, but it's a very wonderful example, by the way. So one organism from the other is different based on the presence and absence of a certain specific sequence of DNA. Like a donkey is different from the cow meat. The donkey is different from the cow because the donkey must carry a certain sequence of DNA in its cells, not every sequence, certain sequences which are specific for donkey. And similarly, the cow must carry certain sequences which are specific for the cow. And we know those specific sequences. So the Food Authority and the researchers should be able to identify whether a meat belongs to donkey or it belongs to a cow or a goat or a mutton. Because the cow meat should carry the specific genes which are only present in the cow. You get the samples, extract the DNA, make specific primers, and see the PCR product. If the PCR product is there, that means the meat is from the cow. If you design a donkey specific primers, which can bind to a target sequence specific for the donkey, in that case, the sample will only amplify if the cells came from the donkey. So with the help of the PCR reaction, you should be able to distinguish between the different organisms. So they can be identified with the help of PCR. I just gave that example because it was quite famous a couple of years back in Lahore. The people were complaining that there is a donkey meat was selling in the streets. So the researchers should be able to find a way how to differentiate between these two. It can also help us to classify the organism. when especially the people who are working with the organism which do not exist anymore. If they found some traces of fossils and bones and they're able to extract some DNA out of it, so they can trace and check and amplify the sequences of DNA to confirm that this bone belongs to that species of a certain organism. We can figure out how close two organisms are based on the similarity of the DNA sequences between them. How close they are. Even in humans and in forensic sciences, people use this ability to check how two people are related to each other. For example, I am 50%, I'm carrying the 50% sequence from my father. So what that means? So my genetical sequences is closely 50% similar to my father. My boy is carrying 50% genetic material from my side. So my boy's genetic material matches 50% to me, but less than 50% to my father. Similarly, me and my brother are carrying 50% similarity with each other because we both are carrying the 50% genetic material from our father. But what about the cousins? If you are related closely, the similarity between the sequences will be high. If you are not related closely, like first cousin, then the second cousin, the similarity will be decreased. So you can figure out how closely the people and the organisms are related to each other. You can distinguish between the species of the organism. They look very similar. Like if you remember, if you see the buffaloes, I'm sure you can get my point. They all look same. They all look similar, but they're not. There are different types of hen out there. They are different from each other. They belong to different strains and different categories, and we can figure it out with the help of PCR diagnosis. We can differentiate between two types of cells. For example, the red one is the cancer cell, for example. Here, the green is the normal cell. The cancer cell should carry the same gene in a different way. A normal cell should carry the same gene in a different way. So a cancer cell is cancerous because the sequence of the DNA is changed, and you can confirm it by different means, and this is the most primitive and most easily detectable way, primitive way, earlier way, easier way to detect the difference between the two genes. So you amplify the sequence of a gene from the cancer cell, you amplify the sequence of the same gene from the normal cell. This is the amplified product from the cancer cell. This is the amplified product from the normal cell. Since both of them are different, they should have a different sequence. Nowadays, we can sequence both of them. But in earlier days, people try to use restriction enzyme. So if the sequence is changed, a restriction enzyme will cut the same piece in a different way, because there might be an extra restriction enzyme site exist here. So this red PCR product will be restricted and cut down into 4 pieces because it has 3 sites, 1, 2, and 3, like this one. It will be visible on the gel in 4 fragments. The green has 2 restriction sites because it's different. It will be digested or restricted into 3 fragments, like this one. So if a same sequence is mutated, is become different, it can be detected based on the difference in their sequences with the help of restriction enzymes. But nowadays we are not using this anymore. We just extract the DNA and we send it for the sequencing. We analyze the sequence. It's as simple as that. We can detect the pathogens like we are doing in case of SARS-CoV-2. If the pathogen is there, in case of SARS-CoV-2, the story does not start from DNA. the story starts from RNA. In case of hepatitis B, the story starts from DNA. So the RNA is converted into DNA, and then we analyze the PCR product. Oh my God, somebody's having a microphone turned on. So वो गलती से लग गया, पकड़ा जाएगा. चलो कुछ सीक्रेट बात कर रहा है. Thank you very much. Okay. हमें लीक हो सकती है बात खतरे में. You can sleep as well comfortably after turning off the things. Okay. So what we are doing in the lab. जी. Anybody wants to ask a question?
जी sir, एक बार please दोबारा बता देते हैं. आप you were telling us about how we distinguish normal cells from cancer cells. आपने कहा primers डिजाइन करके प्राइमर के जो प्रोडक्ट्स आते हैं उसके बाद आपने क्या किया, मुझे एक बार दोबारा बता देंगे.
I was explaining the slide. This is the primitive way. Okay. The primitive way is that if the cancer cell sequence is different from the normal cells, for example, if this cancer cell sequence is different from the normal cells and you are giving them the same restriction enzyme, they will digest the same things in a different way if they are different. The restriction enzyme sites are 3 on the cancer cells, but there are 2 sites on the normal cells. So they cut down the same gene in a different way so we can detect the difference. But this is not the way we are using anymore. What we are doing nowadays, we get the sequence, get the DNA, we send it for the sequencing, and that's all. So in case of SARS-CoV-2, what we are doing, we are getting the samples, extracting the RNA, making it cDNA, and then using the specific primers which are specific to amplify the portions of the viral genome. If the viral RNA is there, the DNA will be there. If the DNA is there, the PCR will be a success. A success PCR will let us know that there exists a virus in the sample. If there is no PCR, we can tell that there is no pathogen present in the sample. Now think about if the kits are not of good quality, if the person who is doing that experiment is not skilled enough, if the person failed to extract the RNA perfectly, अगर किसी ने यहाँ पर गलती कर दी है, if somebody made a mistake in the DNA synthesis here, if somebody made a mistake in doing the PCR reaction, if somebody made a mistake in the analysis, guess 1, 2, 3, 4 quality checkpoints are there. Even the person is carrying the virus. If it is not executed in a skillful way, you might give them the false negative. You might tell them that there is no virus in the person, and that is a huge, huge trouble. I'm having a question from a student. Okay. Oh, that's fine. So, in case if the person is carrying the virus, but the test was executed in an unprofessional way, we might be ended up by telling the person you are all fine. तुम्हारा टेस्ट नेगेटिव है, तो क्या होगा, वो जाके तबाही कर देगा. Are you getting my point? वो खतरनाक तरीन काम. He can start hugging his friends and family, अपने बच्चों के पास रहेगा, and then it's a big trouble. So in case of infectious diseases, that is very important to have lots of control so that you should not make any mistakes. This is an example of the detection of a pathogen. Using 10 picograms, 100 picograms with a different concentrations from the samples. The different concentrations. The more infection means more of the virus. यहाँ पर virus नहीं है. This is an example of the tuberculosis, bacterial DNA. Okay? लेकिन हम suppose करते हैं कि इस पर RNA genome से आया ये. The more RNA, हम suppose कर रहे हैं. This is not an example from RNA. Okay? If more RNA is there, the PCR amplification will be like will produce a lot of PCR product, and that is an indication of the viral load. Lot of virus is there. Then you should have a negative control as well. These are like very less, very na hone ke barabar. So this data will let us know that if you have that much amount of virus, you should get that big PCR product. If you have that much amount of DNA in the sample, you should be able to get that much PCR product. If you have that many amount of DNA in the sample, you will not be able to see any product. And this 12 is a negative control, nothing there. So if you don't have any DNA, you should not see any product. And the 13 is the positive control, that if you manually put the sample out there, if it is there, you should get the product. So positive control is a way to confirm that whatever you are doing is the right thing. Having different templates will let us know that your experimental technique is fine. Having a negative control, having a positive control will confirm that your experiment, you have done the experiment in a wonderful way. So that is how this experiment should be done. This is the same experiment with the protein value. So, sorry, with the PCR value, but I think they took the picture in a different way. So that is how you can confirm, number one, the presence of a pathogen, and number two, how much pathogen is there. Okay, so that's all from my side. It's the floor is open to you and you can ask your question. The first question I am taking from the student. Sir, what do you mean by manually putting a sample? So not manually putting the template DNA. What I was talking is about for this sample, number 13. In number 13, for example, if I am analyzing the SARS-CoV-2 virus, then in one tube, I will put the sequence of the SARS-CoV-2 DNA there. I will put the DNA with the same sequence of the SARS-CoV-2 virus in one of the tube. Not the virus, but the product of the sequence of the virus, viral genome. So I will put a positive DNA as a positive control. I will put the same DNA of the SARS-CoV-2. SARS-CoV-2 is an RNA virus that will be converted into cDNA. And I will put the known sequence in one tube. पक्की बात है वो तो amplify करेगा क्योंकि उसमें हमने खुद से डाला है. That will help us to confirm that whatever we were doing in the other experiment was correct. Because the positive control should give you the result. And the negative control, in the negative control, we do not add any template. We add everything else, but not the template. So if the template is not there, you should not get the product. In a positive control, we put the template. Manually means in khud se dalenge. So if we should get the product here. So now think about an example of a patient. First karte hain. Sample number 8 belongs to the patient. So let's suppose 1 femtogram. This one belongs to the patient. If this is a patient sample, you see the positive control is here. So that means your system is working. You see the negative control is giving no products. That means your system is working. And you see the patient sample is not giving anything. So you are sure that that person is not carrying any infectious. That person is not carrying that DNA. If you are analyzing SARS-CoV-2, you can tell that the person is negative. If this is a person's sample, with the help of negative and positive control, you should be able to check the validity of your experiment, that you are doing the right things. And you also get the product in patient sample, so you can tell with more confidence that the person is positive. Okay. The next question, I'm not taking the names from the people who are sending me the questions in private, okay? अगर आप publicly भेजेंगे तो आपका नाम लूंगा मैं. The next question is that for SARS-CoV-2 diagnosis, are we using simple real-time PCR or quantitative dose viral load matter here? We are doing both of things depending on the question asked by the doctor. The simple PCR is also used to detect the presence and absence of the virus. First thing, the quantitative PCR or a real-time PCR is used to not only detect the presence and absence of the DNA, presence and absence of the viral genome, but it can also help us to quantify the viral load as well. If a person is like, I believe that there is a difference between the price of these two tests. So if you choose to be more specific, more accurate, it should go through the quantitative way. The quantitative way will give you two answers. Number one, the virus is there or not, and if it is there, how much viral load is there. And that will help the doctor to think about his patient in a different way. If the viral load is too much, the doctor should be like proactive and should start worrying a lot. Next question. What is the purpose of negative control? The negative control tells us that if the virus is not there, if the sample DNA is not there, it should not give any product. First karo, आपका सारा का सारा system contaminated है. If your tubes are contaminated, pipettes are contaminated, and you are mixing the things in a wrong way, your negative sample will also give the product. If the system is contaminated, you might get a product. You might get the product from the negative sample as well. Or ये chaos है. It's a chaos. That means your things are not right. The negative sample means you are putting everything other than the template. And if you are getting the product without template, तो इसका मतलब है कोई गड़बड़ है. Your things are not mixed, they are not contaminated or something. और ये भी एक बहुत बड़ा issue है. Some people are saying that even in the negative controls they are getting the positive results. Some people are like they don't have the virus but still they are getting the products. तो ये error है diagnostic expertise का. Okay, next. I received some file. I don't know why. There is a file from one of the students. Anyways, G. If the virus is at the initial stage of infection, would it still be visible on the result of PCR? So it depends on the sensitivity of the PCR mechanism you are using. If you are using very high quality reagents and if you are using very sensitive method, you should be able to detect the virus at the very initial stage. It depends on the amount of money you are spending for a particular test, what type of reagents you are using. What are the expertise of the people who are doing those experiments? You are asking a local lab to do that experiment, or you're asking Aga Khan Hospital or Shokar Khanam to do the same experiment for you. It depends on that thing. एक और मजे की बात है इस सवाल के जवाब में आपको पता होनी चाहिए. Some people who managed to deal with the virus, they got infected like 3 weeks ago, 4 weeks ago. After 3 weeks, they went for the test and they came to know that they are positive. They are not positive, unfortunately, but they are carrying the remnants of the virus in their bodies. Breakdown version of virus, टूटा फूटा virus पड़ा हुआ उनके जिस्म ने मार डाला उसको अच्छे से. But the remnants of the virus are present in the body. Remnant DNA, RNA fragments of the viruses are there. So if you are using so sensitive technique of PCR reaction, that will give you a positive results with those fragments and remnants of the RNA as well. तो एक बहुत सारी एक्सपर्टी चाहिए इस चीजों को फिगर आउट करने के लिए. The next question from Hassan. As we see in coronavirus case, there are many cases in which false results were given. हाँ, अभी बताइए मैंने बात इसकी हसन. Did you got the point? You can answer me in yes or I will repeat my answer. I just answered the same thing. Did you get it, Hassan? You can send a yes to me. हाँ जी फरहान, आपने कुछ पूछना है, आपने हाथ रे. Would you like to speak?
No sir,
जी मैं फरहान एक मिनट में हसन को आंसर कर दूं. The reason why the people are not getting accurate results depends on lots of factors. If you can go back to my slide, this one, हसन अगर आप देख रहे हैं तो I shared here that this is the first step. You need to be perfect here. Second step, you need to be perfect here. Third step, you need to be perfect here. And then the fourth, if any of those steps but not executed in a professional way, you will get the results wrong. And since this is like a new virus, so we are dealing with lots of false results from the labs as well. अभी वो भी उन्होंने भी काम सीखा नहीं है. और एक टेक्निकल वजह मैंने आपको बताई सबसे एंड पे जो बहुत इंपॉर्टेंट है, और वो ये है that in case of many people who manage to successfully fight against the virus, they're fine, but their body contains remnants of the virus. So when they go for the test, जो कहते हैं ना उसको दोबारा टेस्ट किया फिर पॉजिटिव आ गया, दोबारा किया फिर वो बंदा तो ठीक है. If the person is not asymptomatic, the person's body contains remnants of that virus. So the PCR test is showing positive results using the templates DNA from those remnants. So the doctor and the laboratory is saying you are positive, and the person is saying I am fine, and he proves to be fine after a week as well. Sometimes it happens. So all these factors are actually involved in those false results as well. जी फरहान, इससे पहले कि हमारा टाइम खत्म हो जाए, कितना टाइम रह गया?
सर मैं पूछ रहा था कि अगर रेमिनेंट्स भी हों किसी में तो वो फिर भी इन्फेक्ट कर सकता है किसी को?
ओके फरहान, अगर रेमिनेंट्स हो हाईली लाइकली कि वो इन्फेक्ट नहीं करेगा. रिमेंबर इन्फेक्शन क्या होती है, मैंने इस पे एक वीडियो बनाई हुई है, आप देखिएगा यूट्यूब के ऊपर. अगर आपने सब्सक्राइब नहीं किया हुआ तो जरूर कीजिएगा. है फरहान, नहीं तो तुझे छोड़ूंगा नहीं, ऐसे बोलते हैं ना. So I have explained it there कि infection किसको बोलते हैं. Infection is not like a virus is penetrating a cell. A virus is supposed to penetrate into the cell, then it needs to hijack the host cellular system. It needs to be integrated into the host genome, hijack the system, and then start preparing the new viruses. If these stages are not completed, the virus, whatever the virus is doing, it's not infecting. So infection is like producing more viruses out of it. A single mere DNA highly likely is not able to infect in this way. Highly unlikely. Next question. So can we determine the exact region of chromosomal DNA being amplified by the PCR? Of course, we can determine the exact region. And I will be showing you the exact location of every single base pair on human genome. Imagine, human genome, every single base pair, their exact location. Which chromosome, which portion, fragment, subfragment, which exact location. We know everything now. It's very easy. G.